Now let's talk briefly about some of the ways in which we try to enumerate microbes. Uh, how do we know how many cells are actually there? We talked about the population growth curve and the ways in which we can predict how populations will fluctuate over time. But how do we get the numbers of actually how many cells are there? We can't just look at a colony and really get a good estimate of how many cells are there. We can't just look at a tube and see how clotty it is and estimate that there's a billion cells in there. So we want to briefly talk about ways in which we could go about trying to quantify how many cells there and talk about some of the advantages and disadvantages of using one technique over the other. So first, let's talk about ways or methods that are called direct cell counting methods. So the point here is that why would you want to use direct cell count? Well, it's fast, it's quick, and in some cases it's relatively cheap. You can quickly, within minutes, take a sample, a tube, and quantify how many cells are in there. The major problem with it, however, is that it's not going to be able to distinguish between living and dead cells. If there are dead cells in there, if you have a population that's in decline, then it's going to count the dead cells just as evenly as the living cells. And as a result, you would commonly get an overestimation of how many living cells are there. So if your research, if your test is predicated on determining how many living cells there are, then this isn't a good method. These aren't good methods to choose. So one way in which you can use direct cell count would be just to use the microscope. And you can purchase slides that are basically etched with a grid. And these grids are this type of special slide is known as a counting chamber. So you take your sample, you take a small portion of your sample shown here, you pipette it into a cover slip with these grid marks, and then you view it under the microscope. And the instruction sheet that comes along with the slide is going to tell you to count a certain number of grids. So if it tells you to count a 4 by 4 square of grids, then you would count the number of cells. Here, for instance, you count 11, I believe. And then the slide instructions comes with a chart that will, or a table that will allow you to take the number of cells you've counted and then project that into how many total cells there were in your sample. So perhaps it tells you to add a milliliter of fluid onto this slip. You count 11 cells, and then based upon the table, if you have 11 cells in that milliliter, then in a tube of 10 milliliters, then you would have 5 million cells, for instance, just hypothetically. Again, though, dead cells may look exactly the same as living cells. You're not really going to be able to differentiate them because you're going to be killing these cells, of course, using a staining technique. So you don't know which ones were alive before you stained them and which ones were dead, which can be problematic, obviously. Also, you need a lot of cells. You can't just use a sample that has a very few number of cells because that's going to give you widely inaccurate results. Now, something that is a little bit more technologically advanced and a little better than using direct microscopic count is a cell counting instrument. I just want to focus on a flow cytometer because these are types of instruments really used now. Culture counters aren't really used in research any longer. Basically, every hospital has a flow cytometer. They're very expensive machines. And the advantage of using this over direct microscopic count is that it gives you the ability to add the entire sample, to count the entire sample instead of just counting a small fraction of the sample and then trying to estimate the total number. So you take your sample, you throw it into this tube. This tube is connected to a detector, which then will automatically count each time a cell passes through. And for a flow cytometer, the way in which the detector is tripped, if you will, is that there's a laser beam that that is here, and then each time a cell passes through, the laser gets tripped, and then the counter goes up by 1. So again, really fast, really efficient. However, dead cells may be counted in the same way as living cells, so you still have that problem just like you did with direct microscopic counting. So again, let's say your question's a little bit different. Let's say it is important to you that you're able to identify just how many living cells there are in a particular culture. Can you do that? And the answer is yes. The problem is that in order to do that, it's going to take more time. and the methods involved are going to be a little bit more laborious. They're going to be a little bit more difficult and more complicated. So let's talk about 3 ways in which we could try to achieve a viable cell count. One way, probably the most predominantly used way, is to set up what are known as plate counts, which is basically a dilution technique that allows you to estimate how many living cells there were in a culture. And the magic behind this is the fact that if you take a cell that's alive from your sample and you were to throw it on a plate, you would assume that it would then be able to grow on top of this plate, and because it's viable. And if it does grow into a colony, then you can assume that when it was in this culture beforehand, when it was in the tube, it was alive. So based upon that, we can use this information to determine how many cells are living. But the problem is, the obstacle is that, again, in this original culture, there may be 10 billion cells. So even if you take a small fraction of this culture and place it on top of a plate, there's probably going to be too many cells there, and as a result, you're just going to see one big streak, one big slime on top of the plate. There's no way you can individually count the cultures because they've all melted into each other. So you're going to have to also set up what's known as a dilution series. Basically, a setup in which you can dilute the culture to a point in which you can reasonably achieve isolated cultures on top of this plate here. So you typically are going to set up these dilution series in sets of 10. So let's go through the process real quick. You have your culture here, and you have 9 milliliters of what's called diluent. So if you add 1 milliliter, if you transfer 1 milliliter of the culture to this tube, you now have 10 milliliters. And the assumption would be that you've captured 1/10 of the original culture, the original population, and transferred it here. So if there were 10 cells in this culture, and you pick up 1 milliliter of the culture, you would anticipate that you would have picked up 1 cell and transferred it here. And therefore, the total dilution of this tube is 1/10. 1 in 10. Now, still, that's probably not going to be diluted enough to get isolated colonies on your plate. So you're also going to have to continue the dilutions. And to continue it, you're going to take 1 milliliter from here, this first dilution, and then transfer it to a new fresh tube. Again, if you transfer 1 milliliter here, and you had 10 milliliters here originally, now you've transferred 10% of your population. So again, if this was 100 cells, we would assume that we would have transferred 10 cells here, and then 1 cell here. So your total dilution is going to be 1 in 100. And you may continue this process over and over again until you reach dilution numbers of 1 in a million or 1 in a billion. It may take that much to get down to isolated colonies. So the next step is then to take the liquid from here, your dilution series, and transfer a little bit of it onto individual plates. And when you do that, and then when you incubate these plates for a certain amount of time, once you take the plates out, you're going to look for colonies. Now, best practices dictate that you need to find a plate that has between 30 and 300 colonies. So if you can do that, then you're going to successfully estimate the number of living cells here. So an example, let's say you had 100 colonies that form on this plate here. And let's say that when you prepared this plate, you took 1 milliliter from this dilution, this 1 to 1,000 dilution. So you have 100 colonies here. So therefore, if you have 100 colonies here, and you had taken 1 milliliter from this tube, that means that the original population of this tube should be 10 times 100. And therefore, you would expect that there was 1,000 cells in this tube. If there were 1,000 cells in this tube here, then there should be 10 times more here. So there should be 10,000. Based upon the same logic, there should be 100,000 cells here in the first dilution tube, and therefore 1 million cells in the original culture. Now we can also use filtration as a device in order to estimate viable cell counts as well. So we take a filter that has very, very small holes that are so small that liquid will pass through, but the bacteria that are in the culture will get stuck, as shown here. So when you stick all these cells on the filter, you then take the filter and press it on top of a plate, shown here. And any cells that may be alive will grow up into colonies. You count the colonies, and that gives you an estimation of how many cells were in the original culture. Finally, we have MPN technique, which is a viable cell counting technique that's primarily used to determine the potability of water. In other words, how fecally contaminated a water source may be. As I alluded to before, fecal contaminants, that is, organisms that commonly live in the intestines of animals and humans, are commonly gram-negative organisms that like to ferment, that like to produce acids and gas as a result of their metabolism. So, we set up a bunch of tubes that are differential and selective for the presence of gram-negative coliforms. We set up 15 tubes and we add different fractions of your water sample to each tube. Now the key to MPN, if you notice here, is that you have these larger tubes filled with liquid media, but you also have smaller tubes that are inverted, that have been turned upside down. These are commonly referred to as Durham tubes. And the purpose of the Durham tube is to capture any gas that these organisms may be producing. If you didn't have these tubes in here, then the bacteria, if it was producing gas, would make the gas, and then the gas would end up in this top fraction. You would never be able to actually tell whether gas had been produced or not. So we put the little tubes in here, we're going to expect gas to be captured, particularly because these organisms are going to grow at the bottom of this tube, and as a result of that, they're going to be producing gas through fermentation. So ultimately, you add your water samples to these 15 tubes, and then later on, you're going to count the number of tubes that end up getting a bubble. And the more tubes you have that have bubbles, the more fecally contaminated your water source is, and that's estimated using a number called the MPN index. So the higher the MPN, the nastier your water is. Now finally, you can also use biomass to predict how many cells are in a particular culture. And the one biomass measurement I want to focus on here is turbidity. Basically, how cloudy is a solution? How opaque is a solution? And we can measure just how turbid a culture is using something called a spectrophotometer. So a spectrophotometer is going to measure the light transmitted through a sample. Obviously, if we look at this example here, this more turbid culture, because it's cloudier, it's pretty much opaque. We can't see what's behind it. And that indicates the fact that light cannot travel easily through this liquid because it's so cloudy. A lot of light is absorbed by the number of cells in here, the number of the amount of biomass in this tube. Conversely, if there's barely anything growing, the tube remains clear and we can see behind it, meaning that the light is able to travel through without being absorbed. So we can use a spectrophotometer to actually quantify just how much light is actually passing through, and that's shown here. If a lot of light is passing through, then the spec is going to read with a high number, and that indicates that the population is low. If there's a lot of cells, like in this culture, not a lot of light's going to pass through, so you're going to get a low reading on the spec, and that indicates there's a lot of cells. However, like the direct cell counting techniques, like flow cytometry, the problem is that dead cells will absorb light at the same rate as living cells. So that same problem of the fact that you may overestimate the amount of living cells may still be in play.